Okay, I want to welcome you to the 4th Transatlantic ECI GPCR Symposium. On behalf of the organizers, which are Ian and Katrina and Ida and I. They are waving right now. Maybe you can see them. And we're very excited for the next 2 days because we have a schedule packed with science and networking and hopefully also a bit of fun. And we're very happy to have you all here. 30 seconds ago, I said it's 60 people. Now it's 72. We're still multiplying. So as you can see on this first slide, there will be 2 fantastic keynote speakers, Ashish and Maria. And Maria is actually going to kick off the meeting just in a few minutes with the first keynote lecture. And as this is a meeting that focuses on early career investigators, we also have a career panel later in the day. Actually, I think right at the end of the day, featuring 5 panelists from industry and academia and also from publishing. And of course, there will also be a lot of short talks, 18 to be exact, from these submitted abstracts, divided into 5 sessions over the next 2 days. And just to show you what's going to await you, this is a schedule of today. We're right at the beginning with the welcome and opening. And as I said, Maria is going to kick off the meeting with the keynote lecture. And after that, there's going to be a 10-minute SBET session with 2 representatives from SBET. The first short talk session is going to be about computational approaches and structures in GPCR research, followed by the lunch and dinner sessions that have been very much advertised on LinkedIn before, and a poster session afterwards. The day is going to be rounded off with the second short talk session on GPCR signaling and signaling partners, and of course the career panel that I mentioned before. Of course, you are all very welcome to stay on afterwards for networking, just in this Zoom room or in the breakout rooms that we're going to open then. Of course, this meeting would not be possible without our sponsors. The University of Michigan, Department of Pharmacology, the University of Bonn, 70M Antibodies, and SBET. Without you, this meeting wouldn't have been possible, so thank you very much for your support. And as I mentioned before, there's going to be an SBET session where you will hear sorry, a bit more about SBET and also about a special issue in molecular pharmacology that is spotlighting early career investigators, where you can still, until November 1st, submit abstracts. Also, as you can see in the chat, we already posted some organizational remarks. Please stay muted so that the symposium is running smoothly. And also, if it's possible, turn off your cameras during the talk. You're very welcome to have them on when asking questions, and of course also raising your hand and unmuting yourself when called to ask questions during the Q&A. Also, there's going to be a recording of this, and because the sessions are recorded individually, there will be a prompt on the top of your screen, mostly at the beginning of every session. Please just ignore this prompt. And one thing that's important to the speakers is also, if you go to breakout sessions for networking, please always just leave the room and not leave the meeting so that you come back to this room that we share. So again, a very warm welcome. If you have any questions, you can always reach us via email. I think we will also post this in the chat in a minute. Or Tony and Louise, who don't have their cameras on right now, but are patiently in the background, who I want to thank very much for the technical support for this meeting. Thank you very much. So if there's any technical issues, they are also here to help. And with this, I would like to hand over to Katrina and Ida for the first keynote lecture.
Thank you, Judith. Hello to Europe and good morning to Americas, also from our side. I'm Katerina Nemetz, currently working at St. Jude Children's Research Hospital in the group of Madame Babu. Together with Aida Shahraki from University of Marburg, we will be chairing today's first keynote session. There is another member, actually alumni member of Madame's Babu Lab here today, and I believe many of you have joined this session because of her. So please join me in welcoming our keynote speaker, Dr. Marti Solano. Maria is computational biologist and junior group leader at University of Cambridge, the Department of Pharmacology. And as this is an ECI-centered meeting, we ask Maria to share career trajectory with us, and similar, Ashish will do that also tomorrow. So I will be very brief at this point. Maria earned her PhD in biomedicine from University of Barcelona, where she began her exploration of structural computational biology. Then she gained some experience in cheminformatics and virtual screening in the lab of Peter Kolb in University of Marburg. And she joined lab of Madame Babu when he was still at MRC in Cambridge. There, Maria focused on systems pharmacology and data science, exploring how context of natural variation in GPCR isoforms affects signaling, and what implication can this have for drug discovery and development. In 2022, Maria established her own research group at University of Cambridge as a Royal Society University Fellow. Her expertise spans a wide range of disciplines, as I already mentioned throughout the talk, and interestingly, Maria used to work with pipettes too, not just computers. So she has a strong background in structural and systems biology, pharmacology, and I know she's currently interested also in modeling. She's highly skilled and resourceful scientist, kind-hearted and humorous individual, and was therefore an obvious choice for today's keynote. We hope to see more scientists like Maria in the future of academia. Today, we are excited to hear about context-dependent GPCR signaling through the lens of computational biology. Maria, take it away.
Well, thanks a lot for the very kind introduction, Katarina. And yeah, today I wanted to give you a bit of an overview of the work that we do. Can you hear me well? Yes? Okay, perfect. So yeah, I wanted to tell you a bit about the work and the type of work that we do in my group, where we try to combine computational and also experimental data to analyze context-specific GPCR signaling. But first, also following up on what Katarina was saying, my talk will have two different parts. The first part will be about the science that we do in the group, but the organizers also asked me to tell you a bit about my career path and how I managed, or how I ended up being a PI at the University of Cambridge. So the second part of my talk will be about that, and I welcome questions on both parts, basically, at the end of the talk. So first, when it comes to the science, in my group, we are interested in analyzing GPCR signaling complexity. So not only analyzing receptors on their own or by themselves, but also trying to analyze what happens to them in the context of GPCR signaling pathways. And we mainly do it from a data-driven perspective by integrating different sources of biological data, basically. The types of questions we focus on are different. One of them has to do with how structural change translates into signaling diversity, and this structural change, of course, can come from receptors, but also from their transducers, their modulators, and also obviously from their ligands. So what happens with different ligands with different structural properties meet the same receptor? How does that alter the signaling pathway capacity of that receptor? We're obviously also interested in biological context, as we said, as I had said before, and that can be different sources of biological context. It could be the way or the fact that receptors are wired differently depending on the cells that we're looking at or the cell lines that we use to do experiments, but also it can have to do with the context in terms of subcellular context of receptor signaling. And obviously we use different sets of techniques to look at these different sources of variation, basically, that come from biological context. And finally, but not least importantly, obviously, we are interested in why patients respond differently to drugs and how we can use this information to design actually drugs that work better across the board. So we are looking at both changes in patients that have to do with genomics, but have to do with coding, changes in the receptor structure or in the signaling transducer structure that can lead to diversity in GPCR signaling, as well as differences also that might be non-coding and might alter the expression levels of different members of the signaling pathway and what knock-on effects that can have in the way we respond to drugs. So the project I wanted to tell you about today combines a bit of these three questions, and that's why I chose it. And it also it's also interesting because it originated from a network of scientists working on GPCRs. It was the Ernest network, and there was a strong focus in this network in having a systems view at GPCR signaling and trying to understand complex behaviors in GPCR signaling. So in this project, we wanted to analyze actually what happens or how much variation is allowed when you look at GPCR signaling hubs. And I don't think I have to convince this audience that the main suspects in this case would be G proteins themselves, because, let's see, I can show you my pointer. Because obviously there are many, many GPCR signals that will converge in a set of G proteins. Here, of course, we have the 4 G protein families. But if you think of cases like GS, there is not that much choice for receptors that couple to GS to couple to any GS proteins. Obviously, we have the version of the GS. So how much variation would be allowed in a protein like GS? And how can that happen, actually? So what we did basically was look at changes that led to differences in sequence and structure in GS, which were annotated with some physiological phenotypes, let's say, or disease phenotypes, actually. And what you find is 2 sorts of mutations. Some of them are related also to the mutations that will also happen somatically and have to do with gain-of-function mutations in the GS protein that will lead to disease predisposition that is usually related to cancer. But also you have another collection of mutations that are interested, and they generate a spectrum of diseases that have to do with problems with parathyroid hormone receptor signaling. And in particular, we were interested in 2 mutations, which are those 2 on top here.
Okay, sorry. No problem, it's my fault. Okay.
I think you need to mute your microphone. So basically, these 2 mutations were especially interesting to us because obviously I come from a computational structural biology background, and these 2 mutations are in the helix 5 of the G protein. This leucine 399 arginine mutation and also this glutamate 392 lysine mutation happen to be in the helix 5, which we know now that has to undergo actually a disorder-to-order transition for coupling. And also now we know a order-to-disorder transition for G protein dissociation and activation. So how come you can have a mutation in here? And what happens with these mutations? Well, when these mutations were first described by the authors of this paper, and that's more than 10 years ago, they characterized the disease that these patients had. And basically, this pseudo-hypoparathyroidism type 1C is a maternally inherited disease that leads to resistance to the parathyroid hormone. And this leads to problems with calcium resorption and phosphate excretion, actually, in the kidney, that in turn end up leading to a problem in bone formation. And these patients have problems basically with their bone metabolism. And the authors of this paper analyzed these specific mutants, and they saw something interesting. They saw that these mutants don't seem to alter the expression of the G protein, but they do alter the way it behaves. So if you compare wild-type G protein with the rest, what you see is that if you do manage to activate the G protein and then inhibit its inactivation with cholera toxin, you do have a decent level of CAMP accumulation, both for the L388R mutant and for the other one, which actually might lead you to even a higher level of CAMP accumulation than wild type. However, if you try to activate this G protein with a GPCR, and here they are using isoprenaline and the beta-2 adrenergic receptor, the story is very different, because you do see that this mutant basically cannot be activated by the beta-2, while this other one can be activated at the level that would be around 50% of the one of the wild type. They also looked at those responses, and they saw a similar picture. Basically, this is the beta-2 again, activated by isoprenaline. You see the wild type in here. You have an intermediate phenotype with half of the Emax, more or less, for the E392K mutant. And you see a mutation in the mutation L398R that basically doesn't produce any CAMP accumulation. And the same would happen or happened to these authors when they transfected the parathyroid hormone receptor in here, and then they tried to activate the G protein. One of them is basically flat. and the other one is sort of an intermediate phenotype. So what the authors thought, considering that they already knew back in the day that DLX5 was important for coupling, is that probably these mutants are not interacting with the receptor when it's activated by the parathyroid hormone. But of course, now we have much more data. We can start looking at how important these positions are for interaction across GPCRs. So what we first did was take all the GPCRs that we had that were coupled to GS and try to have a look at how important these positions were. And obviously, as you might imagine, they are quite important. So if we use now the common G protein numbering scheme for these positions, we would be looking at positions 24 and 20 in LX5. And if we now look at how frequent interactions are with GPCRs across the board, what you see is that obviously there are hundreds of structures that have an interaction at that point. There are in different parts of the receptor, so that position 24 is interacting mostly with LX6, 7, and 8, especially of the receptors, while position 20 is interacting mostly either with LX3 or with LX5 of the receptor. But obviously, there could be many receptors affected by these mutations in theory. So why are they not? Well, we know already some things about it, but basically the main question is why this receptor is mainly affected. And ideally, we would also like to know with what tools that we have nowadays, and they didn't have 10 years ago, basically, how is signal transduction affected by mutation? And is there something we can do about it? Could we rescue some of these effects with a ligand that is not the parathyroid hormone? So for this first question, as I said, we have to know a bit more about how GS is encoded in the genome. And this is a very, very, very messy picture. You will agree with me. So this is the maternal allele that encodes GS, and this is the paternal allele. And they give you everything you can imagine. So they give you GS, but they also give you an alternative protein in here with an alternative promoter on exon 1 that is actually secreted, doesn't have to do with GS coupling. There are neural versions of GS, both in the maternal and the paternal allele, but the paternal allele also has non-coding RNAs that will modulate the expression of these transcripts. It has the extra long version of GS that you will know probably, and it also has ALEX, which is a very curious protein that comes from an alternative open reading frame from this transcript. And ALEX doesn't have obviously any sequence similarity with the others because it comes from an alternative open reading frame, but it might have some knock-on effects on GS signaling apparently. Above this all, you have these suspicious ends here which stand for methylation, which means that this locus is imprinted in a tissue-specific manner. And this is when it gets interesting for us, obviously, because what happens here is that in some tissues, like the proximal renal tubules, anterior pituitary, the thyroid, or the ovary, you only have expression of the maternal allele, which means basically that our patients who have a mutation on GS on the maternal allele will only rely on their mutated version of GS in these specific tissues, which could explain obviously why they have a phenotype that is restricted to specific contexts in the body. Okay, so now that we know this, and this has been known for a while, obviously, we can go and look at the data we have about receptor coupling that we saw before. This is primary couplings from the guide to pharmacology annotated in GPCR-DB. And then we can go a bit more detailed, or we can be a bit more refined and start looking at what happens if we look at transcriptomics data for GPCR expression in the proximal renal tubules. And now your picture looks different, obviously. You don't have all of the GPCRs that couple to GS expressed in proximal renal tubules. And actually, if you look at the receptor that is the most expressed across the board, here you have your parathyroid hormone receptor. You also have other receptors that couple to GS expressed at a much lower level, and people have not really described that they are implicated in this disease, which could be interesting. But obviously now we understand better why when the parathyroid hormone reaches specifically the kidney and the proximal renal tubule, it will not have this effect on calcium reabsorption, and therefore our patients will have problems in their bone development. So, well, we understand a bit better why the parathyroid hormone receptor is especially affected. What's going on with signal transduction? Well, the first thing we tried to do, and this is a collaboration with a colleague at the Department of Pharmacology, Graham Latz, and this is work from Rosalind Huang, who's a really talented PhD student in his group. She basically tried to replicate these old experiments by looking at CAMP accumulation. So basically what she did is in a GS null cell line, obviously, she transfected different versions of the GS, and then she basically saw a similar picture to what the original paper had seen. This is the wild type. This is the intermediate mutation, E392K. And this is the L388R mutation, where you really have to get to a very high dose, obviously, of the parathyroid hormone to see any CAMP accumulation at all. But we wanted to know a bit more, and of course we have other tools. So we went one step backwards in the signaling pathway, let's say, and we looked at what happens in terms of G protein activation itself. So what we did is we used the TruePath system, and we are basically here monitoring dissociation between the G alpha and the beta gamma in the presence of the parathyroid hormone. And you see a similar picture as you would expect. My light has gone off. So basically, this is the wild type. This is the intermediate mutation, it's 392K, and this is the mutant that basically doesn't signal. And we see a similar picture in here. But then things got a bit surprising, because obviously the authors of the original paper have claimed that the problem actually was that the G protein will not interact with the receptor when you have these mutations present. So we wanted to see exactly that. And for that, we collaborated with the Calaviro lab and Dr. Shannon O'Brien. And what she did is basically she created mini G proteins and monitored how well they interact with the receptor when you activate them, again, with the parathyroid hormone. And here the picture is not the same. So here you have the wild type, and here you have the mutation that doesn't signal at all, L388R. But then here you have the intermediate mutation. And I think you will agree with me that it doesn't interact less than the wild type with the receptor, probably. So what was going on? We wanted to understand this better. And in order to do that, what we tried to do is try to model what was going on with the signaling pathway. And this is work from Theo Redfern-Nichols at the Labs Lab. Theo basically built an ODE-based model of the signaling pathway, where we have the whole activation process of the G protein mediated by the receptor and the ligand. And then what he did is, first of all, try to see whether we could replicate the data for the wild type G protein. So here is the TruePath experiment, but now we have the experimental data here and the simulation data here. And I think you will agree with me that we have quite a nice fit of the data for the wild type receptor. So we do understand what's going on in the signaling system. But then the beauty of it, obviously, is that we can start tweaking things, tweaking properties of the G protein and trying to see which would be the ones that would explain the behavior of the different mutants in this system. And this is what you get. So you can change different parameters of the G protein. And I think one take-home message of this is that actually there are many ways in which you can obviously mess up with the G protein. And you do need your experimental data to inform which of these parameters really make sense in terms of a change. For instance, this KG parameter has to do with the fact or with the behavior of the G protein that leads to binding of the G protein to the receptor. And in theory, as the authors had first claimed, of course, you could reduce the binding of the G protein to the receptor, and you could get both behaviors relatively well. But we do know that the green version of it actually interacts less, but the other one actually interacts more. So that it has to be something else in the G protein that is faulty for this other mutant. And actually, the part that makes more sense for it to be faulty is actually this KGDA parameter, which has to do with how well the receptor will activate the G protein. So it's not about binding anymore. It's about the capacity of the receptor to activate the mutated G protein. And this is the final result, basically. So you can get a nice fit, obviously, of the one that doesn't really couple by reducing 1,000-fold the parameter that explains how well the G protein will bind to the receptor. And by changing only 70-fold the capacity of the receptor to activate this G protein, you can also get a very nice fit for the other mutant. Of course, as I've told you before, I come from a computational structural biology background, so I also wanted to take a structural look. We do have a structure of the parathyroid hormone receptor with GS and an analog of the parathyroid hormone. So the first thing we did was basically introduce these mutations to try to see what might be going on. And for one of the mutants, again, the behavior is probably quite obvious. So we have this L388 position here. This leucine is sitting very happily between LX3 and LX5 in a nice hydrophobic patch. And all of a sudden, if you do try to introduce, of course, an arginine in here, you have a bulkier charged residue that is trying, in theory, to get into this patch. So probably this will never happen. And it can be the reason, obviously, why the G protein doesn't really like to be in there at all. For the other case, it's a bit more subtle because this position obviously is changing charge, but it could still interact with the backbone of LX8 if you have a glutamic acid here or if you have a lysine. So we wanted to understand this better. And for this, we resorted to ND simulations. And these ND simulations were actually run by the Selen lab. As you probably know, the Selen lab is responsible for GPCR MD. And what they did is take the wild type and the mutated system and run some MD to see how stable this interaction actually was. So what we're seeing here is actually the receptor from the bottom, and we're seeing those interactions that happen for at least 75% of the time. So the stable interactions between the receptor and the G protein. And as you can see, for the wild type G protein, you do have a very beautiful conserved groove that goes from LX5, spans LX3, LX2, LX8, up until LX7 in here. For the other mutation, though, you still conserve some interactions, especially with LX3 and LX5, a bit with LX7, but as you see, it is not that stable. The G protein is much more flexible and it's not actually interacting with LX8 or with LX2. So what we think is going on, combining all our data, and this is our model for this, is that actually these two mutations produce the same disease by different mechanisms. So one of the mutations is actually what the authors first claim, preventing interaction between the G protein and the receptor. But the second one still allows interaction of the G protein and the receptor, but it's not a productive interaction. So the receptor or the G protein is activated less efficiently by the receptor, basically, and that leads to a loss of function in this case. So with all we know now and with the models we have, would we find an ideal ligand and a theoretical ligand that could rescue these mutations? And as a bonus track, because we had this model and I knew that Theo was a very talented also PhD student, what would happen in a tissue that had two versions of a G protein, which is what will normally happen elsewhere? So for the first question, of course, I've told you that we analyze the changes in the G protein parameters, but no one prevents you from analyzing changes in ligand-related parameters and seeing what happens to the signaling. So that's exactly what we did. We searched ligand space, and then we tried to find whether there would be a ligand that could rescue the G-alpha activation. And this is what you find. So the rescue ligand is in solid lines. The dotted lines are the original data. And you find that for the E392K mutation, actually, you can get a signaling that is akin of the one of the wild type if you have a ligand with a different alpha parameter. Alpha is a parameter that explains the cooperativity between ligand and receptor, and basically it's something that you can increase by either increasing the K on or decreasing the K off of these ligands. So a ligand that basically would stay longer at the receptor, a long-acting version of the parathyroid hormone, which has actually been postulated as a solution for some of these patients, could work, but it would only work in the case of the E392K mutation. And no matter what you do to the ligand for the other mutation, unfortunately, there's nothing that seems possible pharmacologically. So it is very important, obviously, to know which mutation each patient has in order to see whether you could help them out with a different version of a parathyroid hormone ligand. For the other case, this sounds a bit of a crazy idea of trying to model what happens with two G proteins, but I did ask in particular the Labs lab because I knew that they had run something similar. This is a different paper that has to do with signaling bias. So in this case, they did have a receptor that can couple to two different types of G protein. But of course, if you have a model that tells you about a receptor that can couple to two different types of G protein, nothing prevents you, in theory, from creating a model where you have two different versions of the same G protein, which is exactly what we had in our case. So this is the model that Theo built, where you basically have one wild-type G protein and one either wild-type or mutated G protein in the system with their own parameters that we had found beforehand. And now you can start seeing what happens to the overall activation of G proteins in a particular system that has two different versions together. And we were curious to see what would happen, obviously, because you can start to guess that, well, it is not clear what is worse to have. Is it worse to have a version of a G protein that never interacts with a receptor, but lets the wild-type G protein, do its thing? Or is it worse to have a lousy version of the G protein that still does something, but it might interfere with the way in which the wild-type G protein is doing its work? And the answer, according to our model, is it doesn't matter too much. So basically, if you have a G protein that doesn't do anything, the wild-type G protein will overcome or will take over, basically, and will reach a level of activation that is around 80% of wild-type. If you have the other one, it will still do some of its job, And although it will prevent some of the wild-type G protein from interacting with the receptor, it will still be enough to have around 80% of signaling, which is interesting because it could mean that with this 80% of signaling in tissues that are not affected by the disease, you could actually have a decent level of G protein signaling that would lead to a healthy tissue and a healthy cell function. So with this, I would like to finish this part of the talk. Basically, I hope I have convinced you that combining functional coupling and omics data, oh, sorry about this. Oh my God, okay. Could lead you or could allow you to look at perturbations in the signaling system that are relevant in particular biological contexts, in this case, in the cell type that had this imprinting behavior. And this is something that obviously is not restricted necessarily to the functional interactions between receptors and G proteins. It could be extended to other players of the GPCR signaling system, and it could be extended to any cell type, basically, or any cell line. And this is something that we have been doing in the group. So this is work that we're working on now and finishing with Dr. Shanlin Rao in my group. She's a senior postdoc, and she has been looking at exactly that, what happens at interactions between receptors, G proteins, but also modulators, and their ligands, and how much variability we can expect if we start looking at different systems, which can be different cell types, different cell lines, and what also happens with ligands if we consider that they can obviously be multi-target, both endogenous ligands and drugs. Something else that I hope I have convinced you of is the fact that in some cases we might need orthogonal methods to study loss of function. If we had only studied one endpoint for these mutations, we might reach super different conclusions, obviously, of what was going on. So it can be important to look at different parts of the signaling cycle to understand what's happening in disease, because some loss of function at different points of the signaling cycle can actually result in the same phenotype sometimes. And that this might matter, obviously, for patients, because if we try to treat all the patients without knowing about their mutation with the same solution and the same drug, we might find that it doesn't work just because there is a confounding effect by the fact that the different mutations work in different manners. And finally, that quantifying heterozygosity effects using systems pharmacology can be useful to assess which variants actually result in a phenotype that is important functionally. We have seen that in the tissues that have 2 G protein versions, maybe these mutations are not that important, but of course this could be extended to anything. So we will be sharing this model, and it's already deposited in biomodels, and once the paper comes out, it will be available for everyone to use. But obviously you might think of looking at other sources of variation, for instance, at the level of receptors. So if we imagine polymorphic receptors, and we usually will have heterozygosity, so we will not have 2 polymorphic receptors, but just maybe 1. It might be interesting to look at how many of these polymorphisms actually will result in a change that is significant in the signaling pathway if we can account for these compensatory effects that we have been looking at when we looked at the G proteins, for instance. So with that, I would like to thank everyone who took part in this project. I would like to thank my funders and Ernest, which was the very nice excuse to get it all started. And then I will try to move on a bit into the second part of the talk that has to do with my career path. So Katerina has already given a very nice introduction. There is the first thing, obviously, is just the facts, the timeline. So basically, it all started in Barcelona, where I studied my degree. I studied biomedicine, which was a combination of more cell biology and molecular biology, but also things like anatomy and toxicology and pharmacology. So it was a very nice mix. I was also a bit in the wet lab, as Katerina said. I was working with primary cell cultures. I was doing some work even like with mice. And then I discovered I wasn't particularly good at the lab, and I decided to become computational. That's the truth of it. And then I moved into a computational lab to do my PhD. And there we were using mainly molecular dynamic simulations to understand serotonin receptor signaling bias, and also hallucinogenic effects of serotonin receptor agonists, basically. And from there, I moved on to Germany, to the University of Marburg. And there, my main work was focusing more on virtual screening and trying to find ligands with interesting properties signaling-wise for adrenergic receptors mainly. And from there, I moved to the LMB in Cambridge, where I did more systems pharmacology projects. I was analyzing data from transcriptomics, from structure again, but this time across all GPCRs. And we were looking, as Katerina was saying, at isoforms. And of course, since October 2022, actually, I have my own group at the University of Cambridge. So how did this happen, and did I learn something in the process that could maybe be useful for the rest of you? So first of all, of course, you cannot do it without the support. So I got support in terms of funding, for instance, that allowed me to do it all. So in my PhD, I managed to get 2 PhD fellowships, fortunately, from regional and state sources that supported me during my PhD. I also got some fellowships during my and then, of course, funding from the Royal Society to start my group, actually. Something that was also very useful, of course, was being part of different networks. So in my PhD, I was part of a network in pharmacoinformatics, actually, which was put together 7 different European universities, but also, interestingly, a panel of pharma experts that would come every year and give us feedback on our PhD. It was a bit terrifying, but we learned a lot. So I would recommend these PhD networks. I was also part of GLISEN, which was obviously a cost action related to GPCRs. And that was also very nice because it allowed me to organize a conference together with my PhD supervisor. And in that conference, I actually met my future boss at the University of Marburg. So that was very useful indeed. And I got to know also a lot of people, obviously, in the GPCR community, which convinced me to stay on in this community, actually. And I was also part of EARNEST, which was, of course, the seed for this project we've been talking about. And these days, my group is also part of iGPCRnet, which is also a European international research network on GPCRs. And I attended their last meeting back in the summer, and we've already thought of new ways we could collaborate with them. There were also good news in the path, luckily, and they also help keep you alive in the system. There were prizes, both at the end of my PhD and also a prize from the Biochemical Society, mostly, I think, after I published my Nature paper, obviously, at the LMB. And also I became a member of Wolfson College in Cambridge. That was something called a Junior Research Fellowship, and it was a really nice way of getting into the university and getting to know how it works. Cambridge University is particularly curious and a bit medieval, maybe. So it is nice to know how it works from the inside. But all these parts, basically, you can learn from looking at my CV. So I thought I would also share the fact that, well, scientific careers are messy. And as much as you try to plan them in advance, things will happen in between that will make things more complicated and may force you to reroute your scientific career. And I wanted to show this because this is the part that obviously we don't write on our CV. So basically, seeing how you organize this mess has a lot to do with whether you may stay in academia or not, I think. So I wanted to share some examples of this. So I think the first example of the messy bit has to do with how I organized the conference. And it was a really nice international conference, and that also landed me a postdoc position. But the fact is that one of the main motivators of organizing this conference was that we didn't get funding for the project I was working on during my PhD. So I did have funding because I had managed to get a PhD fellowship, but I didn't have any funding to go to conferences. So basically, that was a very good motivator to organize an international conference, and that was, in the end, good news. But of course, you can imagine that there was some stress in the process. Something else I have told you, of course, is that I got funding to go to the LMB, and it was a really, really nice experience, and I liked it very much. I learned a lot, and it was also very good, obviously, for my career. But one of the motivators from moving from Germany to Cambridge was actually that my partner, who was also a scientist, couldn't find a job in Germany and instead found a job at the University of Cambridge. And that was obviously a way or a motivator to contact the group of Dr. Madame Babu back in the day at the Laboratory of Molecular Biology and moved there for my second postdoc. And again, I have told you that I became a PI at the University of Cambridge thanks to the support of the Royal Society. And of course, I had been thinking about taking the leap, becoming a PI for a while, but there was a big motivator, which was that my research group moved to Memphis, as Katarina will know. So, so that was a bit of a push to apply for transition to independent funding and start my own group, obviously. So the messy bit does exist, and of course, it doesn't appear in your CV. And as scientists, we are very good at taking real life facts and weaving a very nice story out of it. So we are really good at doing that. And all scientists will do that. But you have to be aware that if mess happens in your career, it happens in everyone's career too. And then there are the bad news, and we get bad news constantly as scientists. So there is the funding that I got, but I also tried to apply for funding for my first postdoc that I didn't get, and for extra funding at the LMB that I didn't get either, and also for funding to become an independent PI that I didn't get. And I was also invited to interviews for PI positions that I didn't get. And it continues in these days. PIs don't get funded for some projects very often, unfortunately. So if you do get rejection, think that everyone has a CV like this. And this is part of the take-home messages for this part that I would share with you. And some of them might be useful for some of you too. So everyone in academia has a CV of failures. And this is not my term or invention. You can Google CV of failures and you will find it. We don't write it anywhere, obviously, but it does exist. So don't think that because you do see a failure, that means that your career is not successful. Successful PIs get failure all the time. Getting your own funding can be a lot of work, and it can be frustrating, and it can lead to failure sometimes, but it can also provide a safety net should anything change and the mess happens sometimes. And most importantly, it can give you the intellectual freedom to work on the projects that you really care about. More practically, it also allows you to prove to your future department, if you want a career in academia, that you can attract external funding by yourself. Networking is hard, especially if you're an introvert like myself, but becoming part of networks can help. And these conferences like this can help also. So try to give it a try. The more network you are, the more the easier it becomes, basically, because you do know people in the network already. So it's not a group of strangers that you're approaching every time. Moving from lab to lab is tricky, especially if you're changing countries, and many of you will have changed countries. Takes a lot of energy out of your research also, so you are less productive if you're worrying about non-academic stuff. So do as much research as you can, talk to the people in the lab that you want to join, and talk to the people in the lab that you want to join who already left, and see what they have to tell you, because what works for someone might not work for someone else. So do your research. And my last part would be about mentors. Try to find mentors, but don't find only one if possible. Senior people are great because they have survived the system, they know all the tricks, they have seen it all, but also find mentors that have just taken the leap that you want to take next, because the rules keep changing constantly from the funders, from the institutions. So try to find someone who has survived the system recently and who can tell you all the tips and tricks. And I think with that, I will finish the presentation, and I'm happy to take any questions on any part of the talk.
Thank you very much, Maria. It was great. I believe we had an amazing kickoff of the ECI meeting. It was amazing. So I encourage, we have already 2 questions in the chat. The first one is from Giafei, and since he doesn't have apparently a good mic, I will read it. But Marcel, maybe if you want, unmute after this one, unmute yourself and you can directly ask the question. So the question from Giafei is, great story, Maria. Would these mutations lead to similar effects on different splicing products of the GS gene?
Okay, that's a good question. I guess he means the extra long GS. The thing is that these mutations only occur in the maternal allele, which doesn't have that. In the neural version, it could. The thing is that I don't think the imprinting happens in there. So you would have probably the same situation as in having 2 G proteins, one that is faulty and one that works. So they may happen, I think, but probably it will never happen somewhere where the signaling only relies on these splice versions.
Okay, thank you. And Giafei, you can write another message if you have still follow-ups. And Marcel, I'm not sure. Okay, since I don't see you. Okay, unmuted. I will read it for you. Great talk, Maria, as always. You showed that some G protein mutations have a specific effect on parathyroid hormone receptor. I was wondering whether it would be possible to use the knowledge on the mutational landscape to prospectively design G proteins that are specific for certain receptors. If yes, what do you think which G protein would be the most feasible for that?
G protein specific. I think you could, I guess, design G proteins that are, well, I don't know if specific for a single receptor, but I think you could use the knowledge to do something like that. I don't know about anti-targets, let's say, whether it is possible to say that your G protein will only bind that receptor. Which G protein would be better for that? I don't know. I don't know if GS might be. That's a tricky question, Marcel. Yeah, I need to think about that. Yeah, I think maybe GS wouldn't be a bad protein in the sense that there is not that much overlap as in the other related G proteins like the GIs or the GQs. But yeah, I don't know if people have tried that beforehand. I should probably know that. I think you could, but it would be tricky maybe to have it for a single receptor. I don't know what Marcel wants to do with it, but now I'm curious.
Okay, so I think you can chat later on, or
Yes.
yeah. Is there any other question from the audience? You can either unmute yourself or write it in the chat. But meanwhile, maybe I can ask one, because you mentioned that then you designed a ligand specific for that can overcome this problem of the mutation in the G protein. And I didn't really get how this led to overcoming this issue. Like structurally, did you do MD on this, MD simulations to see how it solves the issue? Yeah.
So basically the ligand, we didn't design an actual ligand. What we found is which properties a ligand should have to overcome the issue, let's say, in terms of basically K on and K off. So what we found is that, it should basically stay for longer in the receptor, and that might be enough for one of the mutations. But we haven't actually produced that ligand. What there are long-acting parathyroid hormone receptor parathyroid hormone versions that are actually available. So that might work actually if you give it a try. It is very tricky to model not real life ligands, but to include all the parameters that come with the ligand in the model directly. So the best way probably would be to try, but we didn't find exactly a ligand that would have these properties that already existed in the arsenal of drugs that target the parathyroid hormone receptor.
Hi, Maria. Thank you for the great talk.
Yeah.
I really
Hi,
enjoyed
Michael.
it. I was wondering, following off what we just were speaking about, would it be possible to design an allosteric modulator or like a molecular glue that could help overcome these effects of either binding or activation through the GPCR?
Interesting. I think for the one that just interacts but doesn't signal so well, probably from the ligand side, you're safer, obviously. Also, we don't understand exactly what's going on with this one in the sense that we don't have structures for all the intermediate states of G proteins binding the receptor. So we think also the first step in which the G protein is recognized by the receptor might also be affected, because the G protein rotates at the beginning and interacts with the helix 6, and that seems to be important. The interaction with this residue seems to be important for that. For the other one, molecular glues, I mean, you might, but the thing is, ideally, you would like some tissue specificity probably, because if not, you might be messing up with all the other GS coupled receptors, potentially, if you're not doing exactly what you... It seems like a challenge because the helix 5 needs to go in, right? So, and the rest of the G protein is exactly the same. So it does, it is really challenging, but it is tricky that for this kind of mutation, there is not much we can think about doing. But yeah, it would be cool. Yeah.
Thank you. And then you were looking at your true path assays, your basically proximity between the G protein and the receptor. Have you looked at other downstream signaling pathways? Are there any that are not changed with the mutant?
The thing is, we haven't looked at things like, for instance, GQ coupling or bit arresting coupling, which would also be interesting, because obviously in that case, it wouldn't be a direct effect, but there might be an interference. So what happens if you really have this mutant that is stuck with the receptor probably, but is not that functional or doesn't get activated so easily with interaction with the other pathways? And how does that translate into problems in the function of the parathyroid hormone receptor? So I think that could be a clear next step, looking at what happens to the rest, just because you have either no G protein interacting in there, which could also mean that the secondary coupling turns into primary coupling, or a G protein interfering in the normal process of PTH1R activation.
Yeah, thank you. That's exactly what I was thinking. Have you seen any differences in internalization?
We have seen that the different mutants, well, the mutant that doesn't interact, it doesn't interact with the receptor in endosomes either. The other one does interact, but because we were using mini-Gs, actually what we see is less internalization, but we think it's about the fact that the mini-G is preventing the normal G protein from promoting the activation cycle and therefore promoting internalization. So we know that the intermediate one does interact with the endosome, but we think it might interact less because the whole activation process might be less in that mutation.
All right, thank you very much.
Cheers.
Okay, thanks. I see another question in the chat, but regarding the second part of your the backstage story that you shared with us. And actually, Gaurav Chudhir asks, what do you do, or how do you motivate yourself when the funding applications are not successful?
It's a tricky one. I know of people who have different solutions. I think a very general one is obviously having people to talk to. So if you have people in the same cohort who are at the same level, hopefully some will be in their high and some will be in their low. So you will compensate each other and be able to help each other out. So I think networks of early PIs, there is a very nice early PI Slack channel, so that I think it's mostly from people in the US, and people are like really helping each other out and just sometimes sharing the bad news, but in a way that helps you out a bit. Then there are people who do things to like cheer themselves up, like for instance, have a good news folder, I know. So every time they get a prize or they get a nice comment, something like that, they put it all together. And the day they are in a crisis, they go back to it and just see all the good news that there are in your life, in your scientific life. And that cheers them up. I think it's also about well, having the whole picture and knowing it's a bit like with social media. You wouldn't think that the life of someone is what you see in their social media posts, right? There is the whole other part of life. In the scientific world, there is sort of the same. So we do show the good parts, obviously, and everyone does the same, and that's the consensus. But that doesn't mean that if you talk to people, you will not see the whole thing. So don't try to be disheartened. If you want to give up at some point, that's also fine. That's the other message. Sometimes you have to put yourself a deadline and say, I will go until here, and then I will find a plan B. And that's also absolutely fine. I think you have to also like have a life and you know how much frustration you can deal with.
Thanks. We still have time for one more question before we move on to the next session. If anyone has a question, just either write in the chat or unmute. Okay. It doesn't seem to be the case. And
I can ask, or I see
okay.
there is question from Ben Lang, which you probably know quite well.
Yes, sure.
He's asking, how did you find that Slack channel?
To tell you the truth, I found the Slack channel because I went to an EMBO group leader course, which is something I would also recommend. And the people in that course were, hi, Ben. The people in that course were some of them were already in it. And yeah, it is nice. It is nice to have that, but it's also nice to have people in your institution, obviously, because they know the actual problems or like advantages that are in your own institution, and that also helps a lot. So I think I would advise both having a wide network of junior PIs if you become a junior PI, but also having a network of people who you can just like visit in that office and yeah, rant a bit with, and that helps a lot, I would say. Cheers, Ben.
I think Katarina wanted to ask something.
Yeah, I will go back to science. Thank you so much for a great talk. So yesterday I was attending a talk from our scientific director, and I learned there are like more than 7,000 rare diseases out there. They are like, and more than 50,000 ultra rare diseases. I know, like you like kind of know you delineated some in GS. I know there are some in GI too. Do you know, like, if there are also like some genetic mutation causing the diseases in other G proteins? And like going back to the question which Marcel had, is there any general way how to attack that? Do you think we could attack that with mRNA or allosteric modulators, like pepdusing, kind of a binding at the interface between specific GPCR and G protein for your case?
I don't know if I know of other I mean, I know of mutations later on in life. So of course, the gain of function mutations also in GQ related to cancer. But I don't know if I know of more. I know of mutations obviously in the receptors themselves, like the ones that cause blindness, for instance, in some GPCRs and so on. But I don't know about G proteins really. I mean, we haven't done, to tell you the truth, we didn't do a comprehensive across the board analysis of that. So that would be interesting to see. My impression about how to find, my impression about how to find
for younger
treatments.
audience.
Yeah, exactly. Yeah. There you go. So my impression would be that it will probably have to be very tailored. So even in the case that we were looking at, there wouldn't be a one-size-fits-all treatment for these patients, I think. So I think you really need to understand the structure and the pharmacology, which is good news for us in a sense. But I don't know if you could really have like across-the-board solution to treat the different mutations. It will have to do, many of the mutations unfortunately have to do with loss of expression, which is not very fancy or not very interesting for us, probably structurally. But they have to do with the dose that you have. That is why it's also nice to have these systems pharmacology models where you can see how much really a dose you need for the different components of the signaling system to have a normal signaling tone. But many of the mutations that are implicated in disease, I think in receptors particularly, are about the receptor not being expressed at all. And then, of course, what you would do is like increase the signaling from that receptor or use a different receptor to activate the same pathway or something like that. But yeah, I think you need to understand the mutation really, really well, probably.
Okay, thank you.
Okay.
We could use some pharmacochaperones, maybe.
Yes.
I believe they are still known for GPCRs out there.
There you go.
Thanks a lot. So let's thank Maria once again for this very wonderful talk. And we can conclude the session now and move on with our schedule. And we have now the ASPET session.